Right, good afternoon, everybody. Welcome to a new session on Life Sciences for the Council of Scientific and Industrial Research National Eligibility Test, along with the Aiims, All India Institute of Medical Sciences, and Neet Biology. I am Dr. Bobby, and I welcome you to our continuous daily lectures on the biological sciences and chemistry on behalf of New Age Cosmetic White EdTech Healthcare Consultancy. I welcome all of you. To start with today's lecture, I wish to start the second chapter of cellular organization, and the topics included under that would be membrane structure and functions, structure of mantle membrane, lipid bilayer and membrane, protein diffusion, osmosis, ion channels, active transport, ion pumps, mechanism of sorting, and regulation of intracellular transport, electrical properties of membranes. B would be structural organization and function of intracellular organelles, cell wall, nucleus, mitochondria, Golgi bodies, lysosomes, endoplasmic reticulum, peroxisomes, plastids, vacuoles, chloroplast, structure and function of cytoskeleton and its role in motility. C is the organization of genes and chromosomes, operon and disruptor genes, gene family, structure of chromatin and chromosomes, unique and repetitive DNA, heterochromatin, euchromatin, transposons, cell division and cell cycle, mitosis and meiosis, their regulation, steps in cell cycle and control of cell cycle, microbial physiology, growth yield, and characteristics, strategies of cell division and stress response. Starting with A1 is the membrane structure. Right? Cell membranes are crucial to the life of the cell. The plasma membrane encloses the cell, defines its boundaries, and maintains the essential differences between the cytosol and the extracellular environment. Inside the cell, the membranes of the endoplasmic reticulum, Golgi apparatus, mitochondria, and other membrane-bounded organelles in eukaryotic cells maintain the characteristic differences between the contents of each organelle and the cytosol. Ion gradients across membranes established by the activities of specialized membrane proteins can be used to synthesize ATP, to drive the transmembrane movement of selected solutes or nerve and muscle cells, to produce and transmit electric signals. In all the cells, the plasma membrane also contains proteins that act as sensors of external signals, allowing the cell to change its behavior in response to environmental cues. These protein sensors or receptors transfer information rather than ions or molecules across the membrane. Despite the differing functions, all biological membranes have a common general structure. Each is a very thin film of lipid and protein molecules held together mainly by non-covalent interactions. Cell membranes are dynamic, fluid structures, and most of these molecules are able to move about in the plane of the membrane. The lipid molecules are arranged as a continuous double layer about 5 nanometers thick. This lipid bilayer provides the basic structure of the membrane and serves as a relatively impermeable barrier to the presence of most water-soluble molecules. Protein molecules dissolved in the lipid bilayer mediate most of other functions of the membrane, transporting specific molecules across it, for example, or catalyzing membrane-associated reactions such as ATP synthesis. In the plasma membrane, some proteins serve as structural links that connect the membrane to the cytoskeleton and or to either the extracellular matrix or an adjacent cell, while others serve as receptors to detect and transduce chemical signals in the cell's environment. As would be expected, cell membranes are asymmetrical structures. The lipid and protein compositions of the outside and inside phases differ from one another in ways that reflect the different functions performed at the two surfaces of the membrane. Membrane ultrastructure and fluid mosaic model. This model, most forcefully proposed by S.J. Schinger and Biochem on the 974, 43, 805, 86 is the currently most favored model. Some consider it the sixth most current modifications of the original Daniel Davison model. It proposes a phospholipid bilayer with the hydrophobic non-polar fatty acid chains directed inwards and the polar hydrophilic heads lining the surfaces. Two would be proteins, glycoproteins, and lipoproteins associated with the inner and outer surfaces of the membrane. The polar portions of the protein associate with the polar surface and may protrude from it, while portions of the proteins may be embedded in the bilayer of lipid. It is proposed that the nonpolar portions of these proteins associate with the nonpolar fatty acid chains. Proteins having both polar and nonpolar area residues are termed amphipathic. Take a good screenshot of this diagram, make a copy and study the details, please. Under physiological conditions, the membrane components are not rigidly fixed but exist in a fluid and dynamic state. The lipids of the membrane are adds fluids at physiological temperatures, thus there may be fluid movement of the lipid molecules and lateral movement diffusion of the proteins that are embedded in the membranes, protein icebergs in a sea of lipids, Singer. This forms a heterogeneous Singer in the sense, courtesy Singer. This forms a high heterogeneous mosaic of proteins which may be continuously rearranged but often with specificity. Number 4, there is not only the lateral mobility of lipids but also exchange from one monolayer of the bileaflet to other monolayers. This exchange flip-flop is much slower than the lateral exchange detected by nuclear resonance studies. This process is facilitated by specific proteins, enzymes called flipases. Proteins may be integral or peripheral, probably most being integral and displaying various different secondary and tertiary forms. The proteins being capable of secondary, tertiary, and quaternary structure changes in response to stimuli may reorient in relation to the phospholipids bilayer. In a phospholipid bilayer, the long fatty acyl side chains in each leaflet are oriented towards one another, forming a hydrophobic core. The polar head groups line both surfaces. The phospholipid bilayer forms the basic structure of all biomembranes, which can also contain proteins, glycoproteins, cholesterol and other steroids, and glycolipids. The presence of specific sets of membranes, proteins, permits each type of membrane to carry out distinctive functions. All cellular membranes line closed compartments and have a systolic and exoplasmic surface and face. The symmetry of biological membranes is reflected in the specific orientation of each type of integral and peripheral membrane protein with respect to the cytosolic and exoplasmic phases. The presence of glycolipids exclusively in the exoplasmic leaflet also contributes to membrane asymmetry. Most integral protein and lipids are laterally mobile in biomembranes. According to the fluid mosaic model, the membrane is viewed as a two-dimensional mosaic of phospholipid and protein molecules. As a phospholipid bilayer is heated, it undergoes a phase transition from gel-like to a more fluid state over a short temperature range. Cholesterol is a major determinant of bilayer fluidity. Although its effect depends on the composition of a membrane, natural biomembranes generally have a fluid-like consistency, and the cells adjust their phospholipid composition to maintain fluid bilayer fluidity. In all cells, proteins in the plasma membrane selectively absorb nutrients, expel waste, and maintain the proper intracellular ionic composition. Proteins in the plasma membrane anchor the membrane to intracellular cytoskeletal fibers and the extracellular matrix or cell wall. In multicellular organisms, plasma membrane proteins also act in the interactions and communication between cells, which are critical for proper functioning of multicellular tissues. In plants, the cell wall, which is built mainly of cellulose, is the major determinant of cell wall and imparts rigidity to cells. Animal cells, which lack a wall, are surrounded by an extracellular matrix consisting of collagen glycoproteins. Now we're on to the second topic, A2, membrane proteins. Transmembrane proteins. Membrane proteins are either extrinsic or intrinsic. Extrinsic membrane proteins are entirely outside of the membrane but are bound to it by weak molecular interactions, ionic, hydrogen, and van der Waals bonds. Intrinsic member proteins, the class we're mainly interested in, are embedded in the membrane. Many of them extend from one side of the membrane to the other and are referred to as transmembrane proteins. Cells are constantly pumping ions in and out through their plasma membranes. In fact, more than half the energy that our bodies consume is used by cells to drive protein pumps in the brain that do nothing else but transport ions across plasma membranes of nerve cells. How can ions be transported across membranes that are effectively impermeable to them? Cells contain proteins that are embedded in the lipid bilayer of their plasma membranes and extend from one side of the membrane to the other. Such transmembrane proteins can function to affect the ion transport in several ways, but how can they cope with the energetically high, unfavorable situation in which an ion must pass through the hydrophobic inner layer of the plasma membrane? Domains. 2 will be domains. If we examine the detailed structure of many transmembrane proteins, we see that they often have 3 different domains, 2 hydrophilic and 1 hydrophobic. A hydrophilic domain consisting of hydrophilic amino acids at the N-terminus is spoken out in the extracellular medium. A hydrophobic domain in the middle of the amino acid chain, often only 20 to 30 amino acids long, is threaded through the plasma membrane, and a hydrophilic domain at the C-terminus protrudes into the cytoplasm. The transmembrane domain, because it is made of amino acids having hydrophobic side chains, exists comfortably in the hydrophobic inner layers of the plasma membrane. Because these transmembrane domains anchor many proteins in the lipid bilayer, these proteins are not free-floating and cannot be isolated and purified biochemically without first dissolving away the lipid bilayer with detergents. Indeed, much of the washing we do in our lives is necessitated by the need to solubilize proteins that are embedded in lipid membranes using detergents. Glycoproteins. Number 3. For reasons that are not well understood, many transmembrane proteins are glycoproteins in the sense that sugar side chains are covalently attached to the hydrophilic domains that protrude into the extracellular membrane. A typical mammalian cell may have several hundred different distinct types of glycoproteins studding its plasma membrane. Each of these glycoproteins will have its extracellular domain glycosylated with a complex branching bush of sugar residues covalently linked to the asparagine side chain. Some glycoproteins may have 2 or 3 asparagine linked sugar side chains, others may have dozens. Multi-membrane spanning proteins. An elaboration of this scheme depicting membrane proteins having single transmembrane domains involves certain membrane proteins that have multiple transmembrane domains. As one scans along the amino acid sequence of these proteins, it becomes apparent that hydrophilic domains, that is, having hydrophilic amino acids, alternate with hydrophobic domains. The protein chain as a whole, when embedded in the plasma membrane, actually waves back and forth between opposite side chains of the plasma membrane. Some think such proteins have the configuration of snakes and hence term them serpentine membrane proteins. A commonly used type of such structure seen in many hundreds of serpentine transmembrane proteins involves 7 hydrophobic domains inserted into the plasma membrane, separated by hydrophilic regions that are looped out alternatively into either the cytoplasm or the extracellular space. A3 would be membrane, transport mechanisms, and ion pumps. Right? All cells acquire the molecules and ions they need from the surrounding extracellular fluid, ECF. There is an unceasing traffic of molecules and ions in and out of the cell through its plasma membranes. Examples: glucose, sodium calcium. In eukaryotic cells, there is also transport in and out of cell membrane-bounded intracellular compartments, such as the nucleus, endoplasmic reticulum, and mitochondria. For examples, proteins, mRNAs, calcium, and ATP. If uncharged solutes are small enough in lipids and lipid-liking, they can move down their concentration gradients directly across the lipid bilayer itself by simple diffusion. Examples of such solutes are ethanol, carbon dioxide, and oxygen. The major difficulty which the membranes pose for some molecule is that the lipid bilayers are not permeable to ions, such as potassium, sodium, calcium, called cations, because when subjected to an electric field, they migrate toward the cathode, the negatively charged electrode. Chlorine and bicarbonate, also called anions, because they migrate toward the anode, the positively charged electrode. Small hydrophilic molecules like glucose and macromolecules like proteins and RNA. But we all know that that cell is intelligent enough to solve the above challenge posed by the membrane. This is how it solves the problem. The passive and active mechanisms move molecules across membrane. Passive transport moves molecules across membranes without expenditure of energy, but cell includes diffusion and facilitated support. To enhance the transport. Active transport uses energy ATP to move molecules across a plasma membrane, include active transport, exocytosis, endocytosis, and pinocytosis. Passive transport. Cell membranes help organisms maintain homeostasis by controlling what substances may enter or leave the cells. Some substances such as water, oxygen, and carbon dioxide can cross a cell membrane without any input of energy by the cell. The movement of such substances across the membrane is known as passive transport. The cell membrane is said to be selectively permeable. And the difference in the concentration of molecules across a space is called a concentration gradient. If these molecules diffusing across the membrane from an area of high concentration to an area of low concentration where water molecules, the process would be called osmosis. Water also moves from a low solute concentration to a high solute concentration. The two types of passive transports are transport by simple diffusion and osmosis, facilitated diffusion, carrier proteins, and ion channels. Diffusion and osmosis. In diffusion, molecules move from higher to lower concentration, that is, down their concentration gradient. A solution contains a solute, usually a solid, and a solvent, usually a liquid. In the case of a dye for diffusing in water, dye is a solute and water is a solvent. Membrane chemical and physical properties allow only a few types of molecules to cross by diffusion. Lipid-soluble molecules, example alcohols, diffused lipids are membranes' main structural components. Gases readily diffuse through lipid bilayer. Movement of oxygen from air sacs alveoli to blood and lung capillaries depends on concentration of oxygen in the alveoli. Osmosis. This is the diffusion of water across a differently permeable membrane from its higher concentration to low concentration. Osmotic pressure is hydrostatic pressure on side of membrane with higher solute concentration produced by water diffusing to that side of membrane. Tonicity is strength of a solution in relation to osmosis. Determinants determines movement of water into or out of cells. Isotonic is where the water relative solution concentration of two solutions are equal. Hypertonic is where relative solute concentration of one solution is less than another solution. And hypertonic is where relative solution concentration of one solution is greater than another solution. Now going to the next page, you can see a graph of simple diffusion and carrier-mediated transport. This is a graph of solute transport across a plasma membrane by simple diffusion. You can see the rate of solute entry increases linearly with the extracellular concentration of the solute. Assuming no change in intracellular concentration, increasing the extracellular concentration increases the gradient that drives solute entry. And in the other graph, we have a graph of solute transport across a plasma membrane by carrier-mediated transport. Here, the rate of transport is much faster than that of simple diffusion and increases linearly as the extracellular solute concentration increases. The increase in transport is limited, however, by the availability of carriers. Once all are occupied by solute, further increases in extracellular concentration have no effect on the rate of transport. A maximum rate of transport, Vmax, is achieved that cannot be exceeded. Now, 2 is facilitated diffusion of ions. Facilitated diffusion of ions takes place through proteins or assemblies of proteins embedded in the plasma membrane. These transmembrane proteins form a water-filled channel through which the ion can pass down its concentration gradient. The transmembrane channels that permit facilitated diffusion can be opened or closed. They are said to be gated. Some types of gated ion channels are ligand-gated ion channels. For that, you could see the figure as ion channels. Ion channels are formed between the polypeptide subunits of integral proteins that span the plasma membrane, providing an aqueous pore through which ions can cross the membrane. Different types of gated mechanisms are used to open and close channels. Ion channels are often selective for a specific ion. Ligand-gated ion channels. Well, many ion channels open or close in response to binding a small signaling molecule or ligands. Some ion channels are gated by extracellular ligands, some by intracellular ligands. In both cases, the ligand is not the substance that is transported when the channel opens. External ligands. These bind to a site on the extracellular side of the channel, for example, acetylcholine, ACH. The binding of the neurotransmitter acetylcholine at certain synapses opens channels that had been sodium and initiate a nerve impulse or muscle contraction. And gamma-aminobutyric acid, GABA, binding of GABA at certain synapses designated GABA, and the central nervous system admits chlorine ions into the cell and inhibits the creation of a nerve impulse. Internal ligands. Internal ligands bind to a site on the channel protein exposed to the cytosol. Examples: second messengers like cyclic AMP, CAMP, and cyclic GMP, CGMP, regulate channels involved in the initiation of impulse and neurons responding to odors and light respectively. ATP is needed to open the channels that allows chloride Cl- and the bicarbonate HCO3 ions out of the cell. This channel is defective in patients with cystic fibrosis. Although the energy liberated by the hydrolysis of ATP is needed to open the channels, this is not an example of active transport. The ions diffuse through the open channel following the concentration gradient. Mechanically gated ion channels. Example: sound waves bending the celiac projections on the hair cells of the inner ear open up ion channels, leading to the creation of nerve impulses that the brain interprets as sound. Mechanical deformation of the cells of stretch receptors opens ion channels, leading to the creation of nerve impulses. Voltage-gated ion channels. In so-called excitable cells like neurons and muscle cells, some channels open or close in response to changes in the charge measured in volts across the plasma membrane. Example: as an impulse passes down a neuron, the reduction in the voltage opens sodium channels in the adjacent portion of the membrane. This allows the influx of sodium into the neuron and thus the continuation of the nerve impulse. So the next will be facilitated diffusion of molecules. Some small hydrophilic organic molecules like sugars can pass through cell membranes by facilitated diffusion. Once again, the process requires transmembrane proteins. In some cases, these, like ion channels, form water-filled pores that enable the molecules to pass in or out of the membrane following its concentration gradient. Examples: maltopore. This homotrimer is this homotrimer in the outer membrane of E. coli forms pores that allow the disaccharide maltose and a few related molecules to diffuse into the cell. The plasma membrane of human red blood cells contain transmembrane proteins that permit the diffusion of glucose from the blood into the cell. Glucose transporters fire different GLUT proteins and two types that co-transport sodium and glucose. These are used for secondary active transport. Water channels, 8 different types of aquaporin transport water across membranes. Note that in all cases of facilitated diffusion through channels, the channels are selective, that is, the structure of the protein admits only certain types of molecules through. Whether all cases of facilitated diffusion of small molecules use channels is yet to be proven. Perhaps some molecules have passed through the membrane by a conformational change in the shape of the transmembrane protein when it binds the molecule to be transported. Active transport. Active transport is the pumping of molecules or ions through a membrane against the concentration gradient it requires. A transmembrane protein, usually a complex of them called a transporter, and energy. The source of this energy is ATP. The energy of ATP may be used directly or indirectly. Direct active transport. Some transporters bind ATP directly and use the energy of its hydrolysis to drive active transport. Indirect active transports. Other transporters use the energy already stored in the gradient of a directly pumped ion. Direct active transport of the ion establishes a concentration gradient. When this is relieved by facilitated diffusion, the energy released can be harnessed to the pumping of some other ions or molecules. Direct active transport. The sodium-potassium ATPase, the cytosol of animal cells, contains a concentration of potassium ions as much as 20 times higher than that in the extracellular fluid. Conversely, the extracellular fluid contains a concentration of sodium ions, sodium plus, as much as 10 times greater than that within the cell. These concentration gradients are established by the active transport of both ions. And in fact, the same transporter called the sodium and potassium ATPase does both jobs. It uses the energy from the hydrolysis of ATP to actively transport 3 sodium ions out of the cell for each 2 potassium ions pumped into the cell. This accomplishes several vital functions. It helps establish a net charge across the plasma membrane, with the interior of the cell being negatively charged with respect to the exterior. This resting potential prepares nerve and muscle cells for their propagation of action potentials, leading to nerve impulses and muscle contraction. The accumulation of sodium ions outside the cell draws water out of the cell and thus enables it to maintain osmotic balance. Otherwise, it would swell and burst from the inward diffusion of water. The gradient of sodium ions is harnessed to provide the energy to run several types of indirect pumps. The crucial role of the sodium-potassium ATPase are reflected in the fact that almost 1/3 of all the energy generated by the mitochondria in animal cells is used just to run this pump. 2. The hydrogen-potassium ATPase, the parietal cells of your stomach, use this pump to secrete gastric juice. These cells transport protons, H+, from a concentration of about 4 by 10 raised to minus 8 m within the cell to a concentration of about 0.15 m in the gastric juice, giving it a pH close to 1. Small wonder that parietal cells are stuffed with mitochondria and uses huge amounts of energy as they carry around this 3 million-fold concentration of proteins. The calcium ATPase of skeletal muscle. In resting skeletal muscles, there is a much higher concentration of calcium ions, calcium Ca2+, in the sarcoplasmic reticulum than in the cytosol. Activation of the muscle fiber allows some of these calcium to pass by facilitated diffusion into the cytosol, where it triggers contraction. After contraction, this calcium is pumped back into the sarcoplasmic reticulum. This is done by a calcium ATPase that uses the energy from each molecule of ATP to pump 2 calcium ions, exchanging from 2 or 3 protons, H+. Pumps 1 to 3 are designated P-type ion transporters because they use the same basic mechanism, a conformational change in the proteins, as they are reversibly phosphorylated by ADP, and all 3 pumps can be made to run backward. That is, if the pump's ions That is, if the pumped ions are allowed to diffuse back through the membrane complex, ATPase can be synthesized from ADP and inorganic phosphate. 4 would be ABC transporters. Right? ABC, ATP binding cassette. Transporters are transmembrane proteins that expose a ligand binding domain at one surface and a ATP binding domain at the other surface. The ligand binding domain is usually restricted to a single type of molecule. The ATP bound to its domain provides the energy to pump the ligands across the membrane. The human genome contains 48 genes for ABC transporters. For example, CFTR, the cystic fibrosis transmembrane conductance regulator, TAP, the transporter associated with antigen processing, the transporter that liver cells use to pump the salts of bile acids out into the bile, ABC transporters that pump chemotherapeutic drugs out of cancer cells, thus reducing the effectiveness. ABC transporters must have evolved early in the history of life. The ATP binding domains in archaea, eubacteria, and eukaryotes all share homologous structure. The ATP binding cassette. The second indirect ATP transport, co-transport by Symport and Antiport systems. Indirect active transport, also termed as co-transport, uses the downward flow of ion to pump some other molecules or ion against its gradient. The driving ions is usually sodium with its gradient established by the sodium-potassium ATPase. Besides ATP-powered pumps, cells have a second discrete class of proteins that import or export ions and small molecules such as glucose and amino acids against a concentration gradient. These proteins use the energy stored in the electrochemical gradient of sodium or hydrogen ions to power the uphill movement of another substance, a process called co-transport. In this diagram, the localization of transport systems to different regions of the plasma membrane epithelial cells of the small intestine is seen. A polarized cell is produced in which entry and exit of solutes such as glucose, amino acids, and sodium occur at opposite sides of the cells. Active entry of glucose and amino acids is restricted to the apical membrane, and exit requires equilibrating carriers located only in the basolateral membrane. For example, glucose enters on SGLT and exits on GLUT. 2. Sodium ions that enter via the apical supporters is pumped out by the sodium potassium ATPase on the basolateral membranes. The result is a net movement of solutes from the luminal side of the cell to the basolateral side, ensuring the effective absorption of glucose, amino acids, and sodium from the intestinal lumen. 1. Symport. In this type of indirect active transport, the driving ion sodium plus and the pumped molecule pass through the transmembrane pump in the same direction. For example, sodium-linked symporters import amino acids and glucose into many animal cells. Many cells, such as those lining the small intestine and the kidney tubules, need to concentrate glucose against a very large concentration gradient. Such cells utilize a 2-sodium 1-glucose symporter, a protein that couples transmembrane movement of 1 glucose molecule to the transport of 2 sodium ions. The sodium-glucose transport is used to actively transport glucose out of the intestine and also out of the kidney tubules and back into the blood. The second example would be that of sodium iodide transporter. This symporter supports iodide ions into the cells of the thyroid gland or the manufacture of thyroxine, and also into cells of the mammary gland to supply the baby's need for iodine. Antiport cells uses movement of an ion across the membrane and down its concentration gradient to power the transport of a second substance uphill against its gradient. In this process, the two substances move across the membrane in opposite directions. This is a diagram of the secondary active transport system. In a Symport system top, the transported solute S is moved in the same direction as the sodium ion. In an antiport system, bottom, the solute is moved in the opposite direction to sodium. Large and small type indicate high and low concentrations respectively of sodium ions in solute. For example, 1. Sodium-linked antiport exports calcium from cardiac muscles. It is one of the best cation antitransporter system. Antiporter system. The plasma membrane of most cells contains one or more types of antiporters which couple movement of a co-transported ion, often sodium, down its electrochemical gradient to move to the movement of a different molecule in the opposite direction against the concentration gradient. In cardiac muscles, for example, a sodium cation plus antiporter rather than a plasma membrane calcium plus ATPase plays the principal role in maintaining a low concentration of calcium 2 plus in the cytosol. It will be interesting to note that the how these two drugs work. The drugs ubiane and digoxin increase the force of heart muscle contraction and are widely used in the treatment of congestive heart failure. The primary effect of these drugs is to inhibit the sodium-potassium ATPase, thereby raising the intracellular sodium concentrations and lowering intracellular potassium, because the sodium-calcium antiporter functions less efficiently with a lower sodium concentration gradient. Fewer calcium ions are exported and the intracellular calcium concentration increases. This increase causes the muscle to contract more strongly. The second example would be of AE1 protein. A chlorine bicarbonate antiporter is crucial to carbon transport by erythrocyte. In addition to cation antiporters, which transport only positive ions, many cells also contain anion transporters, which transport only negative ions. An important example is AE1 protein, the predominant integral protein of the mammalian erythrocyte. This anion antiporter catalyzes the one-for-one exchange of chlorine and bicarbonate across the plasma membrane. Since one singly large charged negative ion is exchanged for another, there is no net movement of electric charge and the reaction is not affected by the membrane potential. Thus, the direction of the reaction is dependent only on the concentration gradients of the transported ions. Transmembrane anion exchange is essential to an important function of the erythrocyte. The transport of waste carbon dioxide CO2, which is generated in peripheral tissues, to the lungs of excretion by respiratory tissues. Let me repeat that again, which is generated in peripheral tissues to the lungs by excretion by respiratory exhalation. Waste carbon dioxide released from cells into the capillary blood diffuses across the erythrocyte membrane. Right? In its gaseous form, carbon dioxide dissolves poorly in aqueous solutions such as the cytosol or blood plasma, but the potent content of the enzyme carbonic anhydrase inside the erythrocyte converts CO2 to the water-soluble bicarbonate anion. The release of oxygen from hemoglobin into the peripheral capillaries induces a conformational change in the globin polypeptide that enables a histidine side chain to bind the proton produced by the carbonic anhydrase reaction. Meanwhile, the bicarbonate formed by carbonic anhydrase is transported out of the erythrocyte in exchange for an entering chlorine via AE1 protein. The overall direction of this anion exchange process is reversed in the lungs. Carbon dioxide diffuses out of the erythrocyte and is eventually expelled in breathing. The lowered concentration of carbon dioxide within the cytosol drives the carbonic anhydrase reaction. As written above, from right to left, HCO3 reacts to yield CO2 and OH. At the same time, oxygen binding to hemoglobin causes a proton to be released from the hemoglobin. The proton combines with the oil to form H2O. The lowered intracellular bicarbonate or HCO3 concentration causes HCO3 to enter the erythrocyte in exchange for chlorine. The next example will be several cotransporters regulate cytosolic pH. The anaerobic metabolism of glucose yields lactic acid, and an anaerobic metabolism yields CO2, which is hydrated by carbonic anhydrase to carbonic acid, H2CO3. These weak acids dissociate, yielding H plus ions, protons. If these protons were not exported from cells, the cytosolic pH would drop precipitously, endangering the cellular functioning. Two types of cotransport proteins are employed to remove some of the excess protons generated during metabolism of animal cells. One is a sodium bicarbonate Cl cotransporter, which imports one sodium ion down its concentration gradient together with one HCO3 in exchange for export of one Cl ion against its concentration gradient. The imported bicarbonate ions combine with the protons generated by metabolism to produce CO2, which diffuses out of the cell. Thus, the overall action of this transporter raises the cytosolic pH, reduces the hydrogen concentration. Also important in removing excess protons is a sodium hydrogen antiporter, which couples entry of one sodium ion into the cell down its concentration gradient to export one hydrogen ion. The plasma membranes of most animal cells contain a sodium-dependent chlorine bicarbonate antiporter similar to the erythrocyte AE1 protein discussed previously. This anion exchange protein functions to lower the cytosolic pH, in effect removing excess OH ions. Recall that a bicarbonate ion can be viewed as a complex of OH and carbon dioxide, so export of HCO3 lowers the cytosolic pH. Exchange of cytosolic bicarbonate for extracellular chlorine is powered by the import of chlorine down the concentration gradient. Cl out is greater than Cl in. The activity of all these antiport proteins depends on pH, providing cells with a fine-tuned mechanism for controlling the cytosolic pH. The proton exporting transporters, which are activated when the pH of the cytosol falls, act to raise the cytosolic pH. Similarly, a rise in pH above 7 stimulates the chlorine bicarbonate antiporter, leading to a more rapid export of bicarbonate and decrease in the cytosolic pH. In this manner, the cytosolic pH of growing cells is maintained very close to pH 7.4. Small changes in the cytosolic pH may have profound effects on the overall cellular metabolic rate. For instance, primary fibroblast cells grown to maximum density, confluence, and tissue culture generally become quiescent, DNA synthesis stops, and the rates of DNA synthesis, glucose catabolism, and protein synthesis are reduced, and the cytosolic pH drops from the characteristic 7.4 of growing cells to pH 7.2. Treatment of quiescent cells with a mixture of serum growth factors re-stimulates cell growth and DNA synthesis. An early effect of these growth factors is a marked increase in the cytosolic pH to 7.4. This dramatic change is caused in part by stimulation of sodium hydrogen antiport, which expels protons into the medium. The rise in cytosolic pH is believed to help activate certain metabolic pathways required for cell growth and division. Example 4. Numerous transport proteins enable plant vacuoles to accumulate metabolites and ions. The lumen of plant vacuoles is much more acidic, pH 3 to 6, than the cytosol, pH 7.5. As mentioned earlier, the vacuolar membrane contains a V-class ATP-powered pump and a unique PPI-powered pump, both of which function to pump H plus ions into the vacuolar lumen against a concentration gradient. The vacuolar membrane also contains Cl and NO3 channels that transport these anions from the cytosol into the vacuole. Entry of anions against the concentration gradients is driven by the inside positive potential generated by the hydrogen pumps. Operation of both of these types of proton pumps in conjunction with these anion channels produces the inside positive electric potential of about 20 MV across the vacuolar membrane and also substantial pH gradient. The proton gradient and electric potential across the plant vacuum membrane are used in much the same way as the sodium gradient and electric potential across the animal cell plasma membrane to power the selective uptake or extrusion of ions and small molecules. In the leaf, for example, excess sucrose generated during photosynthesis in the day is stored in the vacuole. During the night, the stored sucrose moves into the cytoplasm and is metabolized to CO2 and H2O with concomitant generation of ATP from ADP and PI. A proton sucrose antiporter in the vacuolar membrane operates to accumulate sucrose in plant vacuoles. The inward movement of sucrose is powered by the outward movement of hydrogen, which is favored by its concentration gradient. Lumen is greater than cytosol. That is, our concentration gradient is in the lumen is greater than the cytosol. And by the outward negative potential across the vacuolar membrane, uptake of calcium and sodium into the vacuole from the cytosol against the concentration gradients is similarly mediated by proton antiporters. The sodium-potassium ATPase is also an antiport using the energy of ATP to pump sodium out of the cell. K plus in an antiport pump in the vacuole of some plants harnesses the outward facilitated diffusion of proteins themselves pumped into the vacuole by H plus ATPase to the active inward transport of sodium ions. This sodium proton antiport pump enables the plant to sequester sodium ions in its vacuoles. 3. Movement of large molecules. Large molecules, proteins, nucleic acids, polypeptides larger than a few amino acids, polysaccharides larger than a few sugars are not carried by transport proteins. There are mechanisms for moving larger molecules, but they don't enter into the cytoplasm. Endocytosis is a general term for the process whereby very large particles of material are wrapped with plasma membrane and moved into the cell in the form of vesicles or vacuoles. None of the trapped material actually moves through the membrane, but remains on the outside of the original membrane even while the vacuole is inside the cell. Thus, it is defined as a process where a cell takes the macromolecules by forming vesicles derived from the plasma membrane. Vesicle forms from a localized region of plasma membrane that sinks inward, pinches off into the cytoplasm. It is used by cells to incorporate extracellular substances. There are 3 types of endocytosis. A. Pinocytosis. Cell drinking. It is almost the same process as phagocytosis, except it involves liquids instead of solids. During exocytosis, a vacuole containing material to be excreted from the cell moves to the plasma membrane and fuses with it. The vacuole membrane becomes part of the plasma membrane and the contents are released to the outside. Cells use this method to eliminate the waste left after digestion and metabolism and also to release a whole variety of particle materials that have been synthesized inside the cell. Release of hormones and digestive enzymes found in multicellular animals are 2 examples of this process. Phagocytosis is also called as cell eating. Particles are engulfed by phagocytosis, a process that begins when solids make contact with the outer cell surface, triggering the movement of the membrane. The desired particles are then enclosed within a small piece of the plasma membrane, which forms a sac called a vacuole or a vesicle with the food particle inside. This vacuole is then moved into the interior of the cell. Strictly speaking, the food particles are not yet part of the cell as it is surrounded by membrane. Before the food can be used, it must be broken down to smaller pieces and those pieces moved into the cytoplasm. Digestion occurs when the food vacuole is fused with a second vacuole called a lysosome that contains powerful digestive enzymes. Food is degraded, its nutrients are absorbed by the cell, and its waste products are left in the digestive vacuole, which may then leave the cell by exocytosis. Phagocytosis. In the scavenging white blood cells of our body, they prowl around looking for invading bacteria and viruses, which they engulf and destroy them. And receptor-mediated endocytosis. Endocytosis, when coated pits form vesicles when specific ligands bind to receptors on the cell surface. It is a more discriminating process than phagocytosis. Enables cells to acquire bulk quantities of specific substances even if they are in low concentration in extracellular fluid. Example, cholesterol. Membrane embedded proteins with specific receptor sites exposed to the cell's exterior. Cluster and regions called coated pits. A layer of clathrin, a fibrous protein, lines and reinforces the coated pit on the cytoplasmic side. A molecule that binds to a specific receptor site of another molecule is called a ligand. Let me repeat that. A molecule that binds to a specific receptor site of another molecule is called a ligand. Example, iron carried through blood sugar, tightly bound to transparent protein carrier. To get iron into cells, cell membrane contains special receptor proteins that binds transparent, move towards special regions of membranes under which lie clathrin proteins. Endocytosis occurs inside clathrin cage, moves inside cell. Cage eventually recycles back to cell surface, returning the transparent proteins to the external of the cell, or the cell exterior as you may call it. However, iron is released inside the cell, exits from vesicles, becomes bound to ferritin. Exocytosis is the reverse of endocytosis. Quantities of material are expelled from the cell wall without even passing through the membrane as individual molecules. By using the process of endocytosis and exocytosis, some specialized types of cells move large amounts of bulk material into and out of them. It is also called an active transport process. During exocytosis, a vessel moves to the cell membrane, fuses with it, and then releases its contents to the outside of the cell. Vesicles usually budded from ER or Golgi and migrates to plasma membrane. It is mainly used by secretory cells to export products, example insulin in pancreas, or a neurotransmitter from the neuron. The next topic would be A4, mechanism of sorting and regulating of intracellular transport. The protein biosynthetic pathways in cells can be considered to be one large sorting system. Many proteins carry signals outside, but not always a specific sequence of amino acids that directs them to their destination, thus ensuring that they will end up in the appropriate membrane or cell compartment. These sorting systems these signals are a fundamental component of the sorting system. Usually the signal sequences are recognized and interact with the complementary areas of protein that serve as receptors for the proteins that contain them. A major sorting decision is made early in protein biosynthesis, when specific proteins are synthesized either on free or membrane-bound polyribosomes. This results in two sorting branches called the cytosolic branch and the rough endoplasmic reticulum RER branch. As seen in figure 1, this sorting occurs because proteins synthesized on membrane-bound polyribosomes called a signal peptide that mediates their attachment to the membrane of the ER. Further details on the signal peptide are given below. Proteins synthesized on free polyribosomes lack this particular signal peptide and are believed to and are delivered into the cytosol. Then they are directed to mitochondria, the nuclei, and peroxisomes by specific signals, or remain in the cytosol if they lack a signal. Any protein that contains a targeting sequence that is subsequently rebound is designated as a pre-protein. I repeat, any protein that contains a targeted sequence that is subsequently removed is designated as a pre-protein. So here the diagrammatic representation of two branches of protein sorting occurring by synthesis on one systolic proteins listed are encoded by nucleogenes and two membrane-bound polyribosomes. The mitochondrial proteins listed are encoded by nucleogenes. Some of the signals used in further sorting of these proteins are seen in the next list, in the next table. In some cases, a second peptide is also removed, And in that event, the original protein is known as pre-pro protein, example, pre-proalbumin. Figure 2 include many designs destined for various membranes, example of the ER, Golgi apparatus, lysosomes, and plasma membranes. And for secretion, lysosomes are also included. Thus, each protein residue may reside in the membranes or lumens of the ER or follow the major transport route of intracellular protein to the Golgi apparatus. Further signal-mediated sorting of certain protein occurs in the Golgi apparatus, resulting in delivery to lysosomes, membranes of the Golgi apparatus, and other sites. Proteins destined for the plasma membrane or for secretion pass through the Golgi apparatus, but generally are not thought to carry specific sorting signals. They are believed to reach the destination by default. The entire pathway of ER, endoplasmic reticulum, Golgi apparatus,